Hello everyone. Myself, I am Dr. Jitendra from Vignan's Foundation for Science, Technology and Research. And today we are here to discuss about the topic nervous system. So in this chapter, you will be studying about the basics of nervous system, such as how the nervous system is organized, what all are the components of nervous system, what are the functions of those individual components, and what is the classification of nervous system, about the brain, spinal cord, and the nerves. Now let's start with the organization of nervous system. Generally, the purpose of nervous system is it is involved in detection. It detects and responds to the changes that occur within and out of the body. And as we have seen that this nervous system, it consists of brain, spinal cord, and all the nerves which arise from the brain and spinal cord. Nervous system is classified into central nervous system and peripheral nervous system. Central nervous system, it consists of brain and spinal cord, whereas the peripheral nervous system, it consists of all the nerves which arise from the brain and spinal cord. And we can classify this nervous system into central and peripheral. So let's see how this nervous system can be classified. So nervous system can be classified into central nervous system and peripheral nervous system. Central nervous system, it consists of brain and spinal cord, whereas peripheral nervous system, it is categorized into motor and sensory nerves. So motor nervous system involves those nerves which carry impulses from the brain or the spinal cord to the effector organ, and hence these nerves are also called as afferent nerves. Afferent nerves are the nerves which carry impulses from the brain and spinal cord to the organ, whereas the other category of nerves that are present in the peripheral nervous system are sensory nerves. So these sensory nerves, they sense the stimulus or changes in the organ, and this carry the impulses from that particular organ to the brain or to the spinal cord, and hence we can call these nerves as the peripheral nervous system. It consists of two categories. They are motor nerves and sensory nerves. Motor nerves are called as efferent nerves because these nerves, they enter into the organs, whereas those nerves which leave the organs are called as afferent nerves, or these are also the sensory nerves. Motor nerves are efferent nerves, sensory nerves are afferent nerves. The term motor, in the sense they carry impulses from the brain and the spinal cord to the organ, whereas the term sensory, in the sense they sense the impulses or they sense the signals or stimulus from the organs and carry those impulses or signals to the brain and spinal cord. And this efferent motor system, it is categorized into two, that is motor nervous system, or we can say efferent nervous system is categorized into autonomic nervous system and somatic nervous system. Autonomic nervous system, it involves those nerves which are supplied to organs which are under involuntary control, especially these are supplied to the smooth muscles and glands, whereas somatic nervous system, it consists of nerves which are supplied to skeletal muscles or those muscles which are under voluntary control. So in this way, the nervous system, it is categorized into central nervous system and peripheral nervous. Central nervous system consists of brain and spinal cord, whereas peripheral nervous system consists of all the nerves which come from the brain and spinal cord, and these peripheral nervous system can be categorized into somatic efferent nerves and afferent nerves. So efferent nerves in the sense they carry impulses from the brain and spinal cord to the organs, whereas afferent nerves they carry impulses from the organs to the spinal cord and the brain. And this motor nerves or efferent nervous system, it can once again be categorized into autonomic nervous system and somatic nervous system. Autonomic nervous system in the sense those nerves which are supplied to the autonomic organs or the organs which are under involuntary control, whereas somatic nerves are supplied to the organs which are under voluntary control. And this autonomic nervous system can once again be categorized into parasympathetic nervous system and sympathetic nervous system. So it is nothing but the innervation to various organs and what is the influence of that particular nerve stimulation on that particular organ categorizes this parasympathetic nerves and sympathetic nerves. Now you have a close look on what are the central nervous system, what are the parts of peripheral nervous system. So here you can see this brain and spinal cord, it forms a part of central nervous system, and those nerves which carry impulses from the sense organs to the brain and spinal cord, these are called as sensory nerves, or these are afferent in nature, whereas those nerves which carry impulses from the brain and spinal cord to the effector organ, especially skeletal muscles, these are called as efferent nerves or motor nerves. So you can see the sensory receptors which are present in special sense organs such as eye, nose, tongue, skin, ears. So which gives us sensations of sight, hearing, touch, smell and taste. So these are conveyed through these sensory nerves to the brain and spinal cord. Whereas those sensations from internal environment such as chemoreceptors, baroreceptors and proprioceptors are nothing but the receptors of joints and all these things also carry the sensory information through these sensory nerves, and they are supplied to the brain and spinal cord. Now let's see what are the various types of nervous tissue that are present in our body. Generally, the nervous tissue, it consists of two different components or two different types of cells. One is nerves, or which are nothing but neurons, and the other is the supporting tissue which is called as neuroglia. The nervous tissue consists of neurons and neuroglia. So we can broadly categorize these things, neurons and neuroglia, as excitable and non-excitable cells. So we have seen in the unit 1 under basic elements of human body, where we have discussed about different types of tissue. Under that we have discussed about the nervous tissue also, and this nervous tissue, there it is categorized into neurons and neuroglia. Neurons are excitable cells, whereas neuroglia are non-excitable cells. Neurons are involved in transmission of impulses, whereas neuroglia are involved in protection or support of this nervous system. So we will see once again in detail. So here you can see the diagram, and this diagram is nothing but the diagram of an neuron. Unlike other cells or other tissues, neurons cannot divide. They cannot divide. It means that there is no regrowth or formation of new nerve cells. There is only a repair which occurs in this neurons, if at all it is damaged. And these neurons also require oxygen and glucose, and they generate their own ATP, and they have a mechanism of generating impulses and transmitting impulses. and hence these neurons are also called as excitable cells, or which can be stimulated to generate an impulse, which is nothing but an signal. When coming to this structure of neuron, it can be broadly explained that it consists of two parts. One is cell body and the other is all the processes of cell body, or we can call it as extensions of cell body. The cell body's extensions are dendrites and axon. There are many dendrites. You can see there are many dendrites which arise from the cell body, and there is only one axon. The dendrites and axons are the extensions of cell body. This cell body, it forms the gray matter. Wherever the cell bodies are accumulated, they form a gray colored, or we call them as gray matter. Generally, the gray matter, it is present in the peripheral regions of brain. We have seen in the central nervous system, it consists of brain and spinal cord. In the brain, the peripheral regions of the brain consists of cell bodies. It means all the cell bodies, they are present on the peripheral regions of the brain, whereas the internal regions of the brain, it consists of the axons. Whereas in the case of spinal cord, the cell bodies lie in the middle of the spinal cord, whereas in the brain, the cell bodies lie in the peripheral regions, whereas in the middle it consists of axons. In the spinal cord, the cell bodies lie in the middle, whereas the axons lie in the peripheral regions. So the gray matter lies peripherally in the brain, and the gray matter lies centrally in the spinal cord. Next, we will see what a nerve is. So this is the structure of a neuron. A nerve is nothing but it is a bundle of neurons, or it is a bundle of axons of many neurons, is called as a nerve. Whereas neuron can be called as a nerve cell. So we have seen that a nerve cell or a neuron has many dendrites, but it has only one axon. So we will see how the axon is arranged in different types of nerve cells. Generally, the nerve cells are two different types. They are called as myelinated neurons or non-myelinated neurons. Myelinated neurons in the sense they have a myelin sheath on the axon. Hence, these are called as myelinated neurons. Whereas non-myelinated neurons, they do not have such myelin sheath on their axon. The region from which this axon arises from the cell body, it is called as axon hillock, and these axons are very long in length. Sometimes they will be up to 100 centimeters in length, and these axons are intended to carry the impulses from the cell body to the region where they are supplied. And we have seen that there are many dendrites which are attached to the cell body, and in the same way the axonal terminal also, it branches to form different filaments so that they are supplied to different regions of the organs where they are supplied to. Now let's see the structure of a myelinated neuron and a non-myelinated neuron. So in this diagram you can see that axon is surrounded by an sheath, and this axon generally has an outer membrane called as axolemma, whereas that particular sheath which is present or which is wrapped around this axon, it is called as Schwann cell membrane. And this Schwann cell membrane, it consists of fatty substance called as myelin, and hence these nerves which are wrapped with such Schwann cells are called as myelinated neurons. And these Schwann cells are continuously arranged along the length of axon, and they have some gaps between them. In that particular region, the axolemma of axon will be exposed, and hence this particular region where the axolemma is exposed, it is called as nodes of Ranvier. And these myelinated neurons generally are neurons with large axons present in this from the central nervous system, and also the preganglionic neurons are also myelinated neurons, whereas non-myelinated neurons are small nerve cells. Apart from this, even postganglionic neurons are also examples of non-myelinated neurons. Whereas in myelinated neurons, we have seen that the axon is surrounded by an Schwann cell membrane. One axon is surrounded by Schwann cell membrane. Whereas in the case of myelinated neurons, many axons are covered by one Schwann cell, and this does not form concentric layers, and you cannot find any myelin sheath, and the gaps between these Schwann cells also is very less. So that is the difference between myelinated neurons and non-myelinated neurons. So myelinated neurons are having many Schwann cells, and these Schwann cells are enclosed one axon, and there are gaps between these Schwann cells where the axolemma is exposed, and that gaps are called as nodes of Ranvier. Whereas in non-myelinated neurons, many axons of different neurons are enclosed in one Schwann cell, and these Schwann cells, which are arranged continuously, are in very close proximity with each other, so that they won't form any gaps between them, so that the nodes of Ranvier are not visible in this non-myelinated neuron. The axons generally are involved in transmission of impulse from one end of the from the cell body to the end of the axon, whereas the dendrites, they can either receive impulses or they can either transmit impulses. And these dendrites, they form a part of synapse in the case of motor neuron, and in the case of sensory neurons, they are involved in collection of information from various sense organs by acting as sensory receptors. Before going into further details, let us see about various common terms which we will come across in the coming topics. So that is, the first one is presynaptic, synapse, postsynaptic. Generally, we have seen that the nerves transmit impulses, the nerves generate impulses, and the nerves transmit impulses. There is a difference between the generation and transmission of impulses. So if itself generates an impulse, then we can say that the impulse is generated by that particular neuron. And this generated impulse, if it transmits along the length of axon, then we can call that as the impulse is being transmitted. So similar to that of how the electricity flows through the wire, in the same way the impulse also transmits along the length of axon, and once it reaches at the end of the axon or axonal terminal, there it has to transmit the impulse to the adjacent nerve, and in this way the impulse is transmitted from one nerve to the another nerve. And generally that region where this transmission of impulses occur from one region to other region, it is called as an synapse. And this nerve which is present before the synapse, it is called as presynaptic neuron, and that neuron which is present after the synapse, it is called as postsynaptic neuron. Presynaptic neuron, synapse, and postsynaptic neuron. So you can see this diagram where a presynaptic neuron, a synapse, and then postsynaptic neuron are clearly visible. Generally, we have seen that the axonal terminal, it branches into different filaments, and each branch at the end will have this swollen knob-like structures which is called as axonal bulb. And this axonal bulb will come in close proximity with the adjacent cell body, or with the adjacent dendrite, or with the adjacent axon. So wherever it is, they come in close proximity with the adjacent neuron, or in some cases with the adjacent organ. And this particular region where they come in close proximity, and that particular gap which is present between these two neurons, it is called as synaptic cleft. And no two neurons are in direct contact with each other. They are very close with each other, but they are not in direct contact with each other. So we have seen the terms presynaptic, synapse, postsynaptic neuron, and we have seen synaptic knobs or axonal knobs and synaptic cleft. So generally, this transmission of an impulse in the neurons, it will be both electrical and chemical. Electrical in the sense there is a change in the potential or the charge across the axolemma or the axonal membrane, and this change is due to differences in the electric charge or potential between the external environment and the internal environment. And hence this transmission of impulse along the length of an axon will be electrical, whereas the transmission of impulse from one axon to the other axon, it is chemical. And it is chemical because there is a chemical that is released at the end of this axon at the axonal bulb. So generally these chemicals are synthesized by the cell body, and they are stored in synaptic vesicles, and these synaptic vesicles travel across the axon, and they are stored in the axonal bulb or the axonal terminal. Whenever the impulse reaches from the cell body to this axonal terminal, this leads to fusion of this synaptic vesicles with the membrane of the axon, and leads to release of that particular chemicals by exocytosis. So you can see these chemicals are released into the synaptic cleft, and these chemicals will act on the receptors which are present on the adjacent neurons membrane. So we have seen in the unit 1 that there are different types of receptors which are involved in transmission of impulse into the organ, or which are necessary for signaling the cell to function. And hence the transmission of impulse in the nervous system will be both electrical and chemical, and these chemicals are nothing but they are called as the neurotransmitters, because these are involved in transmission of nerve impulse. And we have seen the term somatic nerves and autonomic nerves. And generally this transmission from one neuron to another neuron, we have seen it occurs through chemical mechanisms, and this junction between the two neurons is called as a synapse, and this synapse, it can occur between the axon of one neuron and the axon of another neuron. And this type of synapse, it is called as axonic synapse, or it can occur with the axon of one neuron and the dendrite of another neuron. This is called as axodendritic neuron, synapse. Or this synapse can be between axon of one neuron and the cell body of another neuron, that it is called as axosomatic. Whereas the axon can directly release the chemicals into the extracellular site, that is called as axo-extracellular. Or the axons which are supplied to the blood vessels, they can release the secretions or chemicals into the bloodstream, it is called as axo-secretory. In this way, a synapse between two nerves or between a nerve and the organ can occur in many types: axo-secretory, axo-axonic, axodendritic, axosomatic, and axo-extracellular. Next, we will see what are nerves. We have seen that the neuron is nothing but a single nerve cell, whereas a nerve is a group of nerve cells. A group of nerve cells which are arranged in a bundle, it form one, it is called as one particular nerve. So whenever we talk any nerve, it means that it consists of hundreds of thousands of axons of different nerve cells. And in the previous classes, we have seen how a muscle, especially skeletal muscle, consists of thousands of muscle cells or muscle fibers which are grouped together. There we have seen individual muscle cells are covered by an endomysium, whereas groups of such muscle fibers are once again covered by perimysium, which forms the fascicles, and groups of fascicles are once again covered by another connective tissue membrane which is called as epimysium. In the same way, these neurons also have that particular connective tissue coverings individually or in bundles or as the whole. And that particular connective tissue membrane, which is thin, that covers the axonal membrane, it is called as endoneurium. And such axons bundle or group together to form a bundle, and this bundle is once again covered by an connective tissue membrane which is called as perimysium. And many such bundles are once again grouped together with and covered by an tough connective tissue membrane which is fibrous in nature, that is called as epineurium. Epineurium is the outermost covering of all the bundles, whereas perineurium is the covering of groups of neurons, whereas endoneurium, it is the covering of individual axons. And these bundles of neurons can once again be called as either motor neuron, and we have seen that these bundles of neurons are called as nerves, and based upon the types of nerves that are present in this particular bundles, we can categorize the nerves as motor, sensory, and mixed nerves. Motor nerves, in the sense they carry the axons of those neurons which are involved in transmission of impulse from the brain to the spinal cord. Means in this bundle, it consists of all the neurons which are only involved in transmission of impulse from the brain and spinal cord to the organ, or from the cell body to the axonal terminal. Whereas in the case of sensory neurons, sensory nerves, it consists of axons of all the nerves which are involved in transmission of impulse from the effector organ or from the organ to the spinal cord and the brain. Whereas in the case of mixed neurons, it consists of axons of those nerves which carry motor impulses or which carry sensory impulses. Both these nerves will be present in the one nerve. So these are the three different types of nerves. Now we will see them in detail. Sensory nerves are also called as afferent nerves. You can simply remember that afferent, A, in the sense they carry impulses away from the organ. Organs, generally they transmit only sensory impulses. And what are various regions where these sensory impulses are collected can be categorized broadly into four types of regions. They are somatic, cutaneous, and common senses. Generally, these are senses which arise from the skin or from the such as pain, heat, cold, touch, all are the somatic senses or cutaneous senses. The next is proprioceptor senses. So these are the senses which originate from the muscles and joints, and based upon these sensory impulses from joints and muscles, the brain can know what is our position, and it will send respective sensory motor impulses to correct our position. So unless we know how our joint is, how our muscles are, we cannot correct or rectify our posture. And this sensation of impulses from these muscles and joints will be carried by this sensory nerves to the spinal cord, and from there to the brain. The other type of senses are from special sense organs such as eyes, ears, nose, and mouth, where we can get the impulses which are generated by the vision, by hearing, by tasting, and by smelling. Whereas there are sensory nerves which carry impulses from the effector organs which are not under voluntary control. That means those organs which are not under voluntary control are having autonomic afferent nerves, means they carry impulses from various chemoreceptors, various baroreceptors. Chemoreceptors, generally they are involved in sensation of chemical changes within the blood, whereas baroreceptors are involved in carrying impulses of pressure changes within the blood. The blood pressure changes are sensed by this baroreceptors, whereas the pH and all are sensed by this chemoreceptors. And all the organs, glands, tissues which cannot be controlled by us, they send impulses through this autonomic afferent nerve. And the other type of nerves are motor nerves. Motor nerves are also called as efferent nerves. So to simply remember, you can recall them as efferent, E, in the sense those nerves which are entering into the organ. Means these nerves, they carry impulses from the brain and the spinal cord to the effector organ. So the impulse will transmit and it will enter into the organ. And this is called as motor nerves or efferent nerves. And these motor nerves, they generally originate from the brain or from the spinal cord or from the autonomic ganglia. Autonomic ganglia are nothing but cell bodies which are present in groups are called as autonomic ganglia, and these cell bodies which are present outside the central nervous system, they are called as ganglia. If the groups of cell bodies are present outside the central nervous system, they are called as ganglia. Except those groups of cell bodies which are present within the central nervous system, where they are called as nuclei, except basal ganglia. So which is nothing but a group of cell bodies, but it is present within the brain. And hence all the motor neurons, they originate from either brain or from the spinal cord or from the autonomic ganglia. And these motor neurons are of two different types. Those neurons which carry impulses to the skeletal muscle, they are called as somatic motor neurons or somatic nerves. And those neurons which carry impulses to the autonomic organs, these are called as autonomic motor neurons. And these examples are sympathetic and parasympathetic nerves, which are involved in transmission of impulses to various smooth muscles or to various glands, cardiac muscles, etc. And we have seen that there are various mixed nerves which consists of the axons of both motor neurons and sensory neurons. So generally we can see these mixed nerves at outside the spinal cord. So outside the spinal cord, these spinal nerves will have both the axons of motor neurons which are coming from the spinal cord and those axons of sensory neurons which are entering into the spinal cord. So in this way, the mixed nerves, they consist of axons of both motor nerves and sensory nerves. Now let's see about what are various nerves, what are various neurotransmitters, what is a synapse, what is a presynaptic neuron, what is a postsynaptic neuron, what is a ganglia. So in the first case, you can see that is a somatic nerve, and this is an example of somatic motor nerve. It means the impulse generates in the spinal cord and this impulse is transmitted across the axon, and this axon is supplied to an effector organ which is nothing but an skeletal muscle, and the neurotransmitter that is released at the end of axon is called as acetylcholine. So acetylcholine is the neurotransmitter that is released at the end of an somatic neuron. In the next case, we will see about autonomic neurons. Autonomic motor neurons are once again of two different types. We have seen that they are sympathetic neurons and parasympathetic neurons. So sympathetic neurons, they originate from the spinal cord and they leave the spinal cord as presynaptic neuron, and this presynaptic neuron, it forms a synapse with the cell bodies that are present outside the central nervous system, and they are called as sympathetic ganglia. So this neuron starts from the spinal cord and it ends at the ganglia. So the neurotransmitter that is released at the end of this presynaptic neuron is acetylcholine. And once the acetylcholine is released, it acts on the cell body which is present in the ganglia, and this stimulates that particular cell body. Once the cell body is stimulated, this once again generates an impulse, and this once again leads to transmission of impulse. And this neuron which arises from the ganglia, it is called as postganglionic neuron. Preganglionic neuron, ganglia, and postganglionic neuron. And the end of this postganglionic neuron will release a chemical. Here it is called as noradrenaline. So the sympathetic motor efferent neuron, it originates from a spinal cord as a presynaptic neuron, ends at the ganglia, and from the ganglia another neuron it is originated. This is called as postsynaptic neuron, and the neurotransmitter that is released by the presynaptic neuron at the ganglia is acetylcholine, and that neurotransmitter that is released by the postsynaptic neuron at the end or the effector organ, it is called as noradrenaline. In the last, you can find parasympathetic motor neurons. So parasympathetic motor neurons generally are originating from the spinal cord, and the presynaptic neuron once again is called as preganglionic neuron. It releases acetylcholine at the ganglia, and that postganglionic neuron which is originated from the ganglia, once again it releases acetylcholine at the end. So here the neurotransmitter that is released both by the preganglionic neuron and the postganglionic neuron is acetylcholine in the case of parasympathetic nerves, whereas that neurotransmitters in the case of sympathetic nerve, the preganglionic neuron will secrete acetylcholine, whereas the postganglionic neuron will secrete noradrenaline. So this is we have seen regarding the nerves. And in the nervous tissue, we have seen that there are other supporting cells which are called as neuroglia. and this we have once again discussed them previously in unit 1, where this neuroglia are also called as glial cells, are of 4 different types, and they are involved in supporting this nervous tissue. We will see them once again. The 4 different types of neuroglial cells are called as oligodendrocytes, ependymal cells, microglia, and astrocytes are the 4 different types of neuroglial cells. Oligodendrocytes, ependymal cells, microglia, and astrocytes. Oligodendrocytes, generally they are found in clusters and they are present around the nerve cell bodies, especially in the gray matter, and they are also present along the axons of myelinated fibers, and generally these are essential for maintaining the myelin content in these regions in the same way as the Schwann cells regulate the myelin in the peripheral nerves. The peripheral nerves are once again wrapped with a Schwann cell which consists of myelin sheath. In the same way, these oligodendrocytes are present in the cell bodies and also along these myelinated neurons in the central nervous system, and they maintain the myelin levels. Other type of cells are ependymal cells, and these ependymal cells, they form the epithelial lining of the ventricles of the brain. We will come across this term, ventricles of the brain, and they form the lining of these ventricles of the brain, and these are involved in secretion of cerebrospinal fluid. The other type of cells are microglial cells, and these microglial cells, they are the smallest in size and also they are least in number, and these also are the monocytes which are migrated to the central nervous system. They get differentiated into microglia, and they are having the same function as that of monocytes, which are phagocytotic in nature. So they are involved in phagocytosis of any microbes or damaged tissues. And the other type of cells are astrocytes. We have seen that these astrocytes, they are the main supporting tissue and they are star shaped in nature. And some astrocytes at their processes, they have an elongated, or we can say swollen foot process, and this swollen foot process, it forms a covering against the blood vessels that are supplied to the central nervous system, especially brain. The foot process forms another lining on this blood vessels, and these blood vessels also are having epithelial tissues, and these blood vessels also have endothelial cells which are very tightly attached with each other, and this is once again formed by another covering of foot processes of astrocytes in this way, so that they form 2 layers, and these 2 layers together they form the blood brain barrier, and this barrier will restrict the transport of substances across this barrier from the blood into the brain. Generally, it won't allow any toxic substances or water soluble substances to enter into the brain. Only lipid soluble substances, glucose, oxygen, they can cross this blood brain barrier. In this way, this blood brain barrier will protect the brain from harmful substances. Next, we will see what are various membranes which cover the various parts of central nervous system. We have seen that central nervous system, it consists of brain and spinal cord, and this brain and spinal cord are covered by various membranes which are called as meninges, and these meninges are of 3 different types, or there are 3 different membranes which cover the brain and spinal cord, of which the outermost membrane, it is called as dura mater, and the middle, it is called as arachnoid matter, and the inner, it is called as pia mater. Dura mater, arachnoid matter, and pia mater. Now we will see them in detail. Generally, they are arranged in this way: the outer dura mater followed by a subdural space, or the space between the first membrane and the second membrane. It is called as subdural space, followed by arachnoid matter, followed by subarachnoid space, and followed by pia mater. Now let's see them in detail. The dura mater is the outermost membrane, and this dura mater it composed of two layers. The outermost layer it replaces the periosteum of the skull, whereas the inner layer is the protective covering of the brain, and this inner layer it sweeps between the cerebral hemispheres, which lead to formation of flux cerebri, and it sweeps between the cerebrum and cerebellum, which it forms the flux tentorium cerebelli, and between the hemispheres or lobes of cerebellum to form flux cerebelli. So in this way, this diagram shows how the inner layer of dura mater it folds to form flux cerebri between the lobes of cerebrum, and flux cerebelli between the lobes of cerebellum, and flux and tentorium cerebelli between the cerebrum and cerebellum. And generally venous return of blood from the brain, and generally venous blood of the brain also enters into the sinuses that are formed between these two layers, and generally this dura mater it forms the superior sagittal sinus, which is found between the lobes of cerebellum, that is the flux between the lobes of cerebrum, that is flux cerebri, and it also forms the straight and transverse sinuses between the tentorium in the lobes of cerebrum and cerebellum. Whereas the spinal dura mater it is a single layer. Generally it originates from the foramen magnum and it ends at the second sacral vertebrae. And once again the outermost layer it lines the vertebral canal of vertebrae. The spinal nerves and the blood vessels, spinal nerves enter or leave the spinal cord through the epidural space that is present between the inner dura mater and the outer dura mater. The arachnoid matter it is a fibrous tissue and which is present between the dura mater and the pia mater. So it is separated from the dura mater by a subdural space, so which consists of serous fluid, and this is separated from the pia mater by a subarachnoid space which consists of cerebrospinal fluid. The arachnoid matter passes over the convolutions of the brain and it accompanies the inner layer of the dura mater in the formation of flux cerebri or tentorium cerebelli or flux cerebelli. And it also continues downwards to cover the spinal cord, and it also ends up by merging with the dura mater at the level of second sacral vertebrae. The third layer is pia mater, and this pia mater it is adhered to the brain and spinal cord. So this is a very delicate layer of connective tissue. So all the three layers of membranes or meninges are connective tissue membranes, whereas pia mater is a delicate layer of connective tissue and it contains many minute blood vessels, and we have seen that it is adhered to the brain completely, covering all the convolutions or depressions into each fissure or gaps between the lobes. Everywhere this pia mater is adhered and it is entering, whereas we cannot see that arachnoid matter and dura mater it is adhered or it is directly attached to all the depressions or to all the fissures of brain. And this also it is continuous downwards surrounding the spinal cord, once again attaching to the spinal cord, and after that generally it continues as a single spike like structure which is called as phylum terminale, and this phylum terminale, it pierces the arachnoid tube and goes on and fuses with the dura mater at the periosteum of coccyx. Now let's see about the ventricles of the brain. We have seen the meninges of the brain. Meninges are the membranes which cover the brain and spinal cord. They are protective in nature. Whereas the ventricles of the brain are hollow cavities which are present within the brain and spinal cord. They are 4 in number. First and second are right and left lateral ventricles, and the third one is third ventricle, and the fourth one is fourth ventricle. Right and left lateral ventricles, third ventricle, and the fourth ventricle. Now this right and left lateral ventricles, they lie below the cerebral hemispheres. The cerebrum, it consists of 2 hemispheres, and below these cerebral hemispheres they are the right and left lateral ventricles, and this are present just below the corpus callosum. Corpus callosum is nothing but this consists of bundles of axons of the cell bodies which are present in the peripheral regions. And this lateral ventricles, they communicate with the third ventricle by interventricular foramina. So all the ventricles of the brain, they are connected with each other. The right and left lateral ventricles are connected with the third ventricle by interventricular foramina. The third ventricle, it is present below these lateral ventricles, and it is present between the thalamus. It continues or it communicates with the fourth ventricle by cerebral aqueduct. Whereas the fourth ventricle, it is present below and it is present behind the third ventricle, and it is present between the pons and the cerebellum, and it continues with the central canal of spinal cord and communicates with the subarachnoid space by a foramen that is present on the roof of fourth ventricle. So we have seen that the ependymal cells which are lined or which line this ventricles, they secrete cerebrospinal fluid, and this cerebrospinal fluid also it enters into the subarachnoid space by the pores which are present on the roof of this fourth ventricle, so that cerebrospinal fluid that is secreted into the ventricles, it enters into the subarachnoid space of the meninges. And we have seen that the subarachnoid space consists of cerebrospinal fluid, and we also have seen that the subdural space consists of serous fluid. Now let's see the cerebrospinal fluid. So here in this diagram you can see how the right and left ventricles are connected with the third ventricle and how it is connected with the fourth ventricle. You can see how cerebrospinal fluid is secreted into the right and left ventricle. From there it passes into the third ventricle through the interventricular foramina, and from there into the fourth ventricle by the cerebral aqueduct. And at the roof of this fourth ventricle you can see that there are pores through which the cerebrospinal fluid, it is entering into the subarachnoid space. So once it enters into the subarachnoid space, it circulates throughout the brain and spinal cord in this subarachnoid space. And the cerebrospinal fluid, once again, it enters into the bloodstream through arachnoid villi that is present at the roof of the brain. There you can see at the roof of the brain you can find superior sagittal sinus. So that is the region where arachnoid villi are present, and this arachnoid villi, they act as valves, one-sided valves, so that the cerebrospinal fluid can only go out of this subarachnoid space, but it cannot come in. And this opening of the valve depends upon the pressure of cerebrospinal fluid, and an approximate of 0.5 ml of cerebrospinal fluid will be secreted for every minute, making up to about a total of 720 ml per day, and most of these things will be entering into the bloodstream through the arachnoid villi, and approximately 150 ml of this cerebrospinal fluid will be present in the subarachnoid space. Now let's see what is the composition of this cerebrospinal fluid. Cerebrospinal fluid, it consists of water, mineral salts, glucose, plasma proteins such as albumin and globulin, leukocytes, creatinine and urea. And let's see the functions of this cerebrospinal fluid. Cerebrospinal fluid, it supports or it protects the brain and spinal cord by acting as a cushion, and it acts as a shock absorber. And it also keeps the brain and spinal cord moist, and it is also essential for exchange of nutrients from the cerebrospinal fluid into the tissues of brain and spinal cord, and removal of waste products from the brain and spinal cord. And this is also necessary for maintaining the respiratory activity, because cerebrospinal fluid, it covers the medulla oblongata, which consists of various chemoreceptors which are necessary for sensation of oxygen levels within the cerebrospinal fluid. Once these chemoreceptors sense any abnormality in the oxygen levels of cerebrospinal fluid, it leads to stimulation of respiratory center within the medulla, and it enhances the respiration. Now we will see about the various parts of central nervous system, that is the brain and spinal cord. First we will study about the brain. Brain, it is the largest. Brain, it is one of the large organs which weighs approximately 1.4 kilograms, and it is enclosed in the cranial cavity. And this brain, it consists of various parts. They are the cerebrum, diencephalon, brainstem, medulla oblongata and cerebellum. We know cerebrum or cerebral cortex, whereas diencephalon, it consists of thalamus and hypothalamus, whereas the brainstem, it consists of midbrain and pons, which is followed by medulla oblongata, and it also consists of cerebellum. And this brain, it is supplied by an artery called as circulus arteriosus, which is the artery which is round in arrangement, and it supplies to the brain. And approximately 15% of the cardiac output is supplied to the brain through this circulus arteriosus, whereas the venous blood from the brain, it is collected into the various sinuses that we have seen previously. They are superior sagittal sinus, straight sinus and transverse sinuses. So in this diagram you can see the cerebrum, you can see the thalamus, hypothalamus, which forms the diencephalon, and you can see the brainstem, which consists of midbrain, pons, medulla oblongata and the cerebellum, so followed by the spinal cord. And this is a mid-sagittal section of the brain. They are the frontal lobe, the parietal lobe, the occipital lobe and the temporal lobe. So based upon the regions of the cranial bones where they are present, the regions of brain also are categorized into various lobes: frontal lobe, parietal lobe, temporal lobe and occipital lobe are the different lobes. And when in this longitudinal section, you can see that there are various depressions, and there are minute depressions and there are major depressions. That minute depressions are called as sulci, whereas that major depressions are called as fissures. You can see that the two cerebral hemispheres, they have an longitudinal fissure in the middle, whereas the whole cerebrum, it consists of various depressions called as sulci, and that particular region which is present outside, it is called as gyri. And we also have seen that all the cell bodies of this brain are present in the peripheral regions which form the white matter, whereas all the axons of these cell bodies, they are present in the middle which form the white matter. And we can also see various regions within the brain, that is the thalamus, basal ganglia, hypothalamus and all, which lie within the brain. So now we have seen what the brain consists of. So brain, generally it is involved in various higher functions, and all the essential arrangements within the brain are present in the cerebral hemispheres, and the axons of all these cell bodies which are present in the cerebral hemispheres are present in the middle, whereas here these nerves are formed into bundles and they are called as tracts. The nerve bundles which are present within the brain, these are called as tracts. There are three different types of tracts which can be seen within the brain. They are association tracts, commissural tracts and projection tracts. Association tracts, commissural tracts and projection tracts. So here association tracts are many in number and they connect with different parts of cerebral hemispheres from one gyrus to another gyrus. We have seen that there are many depressions like this, and this exposed region, it is called as gyrus. Association tracts are many in number, so that they connect with one gyrus to the other gyrus, one gyrus to the other gyrus. Since there are many gyri which are arranged like this, so all these gyri are interconnected by these association tracts. Whereas commissural tracts, they connect with corresponding areas of two hemispheres, from the right hemisphere and the left hemisphere. So these commissural tracts, they correspond with each other from this region, from this region. So both the hemispheres are present at the same regions, or they are involved in the same function. Example of this commissural tract is corpus callosum, and the other tracts are projection tracts. So these projection tracts which arise from the cerebral cortex, they are involved in association with the lower parts of the brain and also the spinal cord. And this example of this projection tract is internal capsule. So internal capsule is nothing but those nerve tracts which are connected or which communicate between the cortex and the basal ganglia, cortex and thalamus, and these carry impulses both to and from the cortex, and that motor fibers of internal capsule, they are called as pyramidal tracts. So projection tracts, example is internal capsule, and this which are involved in motor impulses from the cortex, they are called as pyramidal tracts. And these pyramidal tracts, they undergo decussation or crossing over at the region of medulla oblongata. And those motor nerve tracts which do not pass through this internal capsule, they are called as extrapyramidal tracts, and these are involved in transmitting impulses to the basal ganglia.